Good evening. Good evening, everyone. Hi. I'm Pankaj Shah. I'm the director of the Queensland Brain Institute. And look, welcome everyone post Alfred. And I'm glad to see everyone survived. And look, welcome to Brain Awareness Week and the discussion today. And let me start by acknowledging the Tuggerah and the Jagera people, the custodian owners of this land in which we're on today, and pay respect to their elders, past and present, and they will have looked after this land for many, many years, and at least some of them, I'm sure, will continue to do so if we keep listening to people. So look, this is Brain Awareness Week, and our brains, it's all about our brains, and our brains are an organ in our body, just like every other organ, and it sits inside here, so it weighs just over 1 kilo, and really it makes you the people that you are, the persons that you are, and it's responsible for everything that you do. You know, how you move around, your behavior, your thoughts, your memories, and how you like things or don't like things. And of course, like every other organ, it has its problems. You know, it can get trauma and it can be diseased. And diseases of the brain lead to a host of problems in the community and with us, and with some of which are recognized and some which are not recognized that well. And they account for nearly 50% of the burden of disease in our community. And really, unlike so many other organ disorders like cardiovascular disease, you know, if you have a heart attack, you know, you can go to the hospital and be fixed pretty quickly these days. We understand very little about the brain. And really, the QBI, the Queensland Brain Institute, was set up pretty much to try and understand what goes on in the brain. And of course, one of the biggest disorders, as we are living longer and longer now, are neurodegenerative disorders. These are disorders which come along as we age. And, you know, if we all died at the age of 50, which perhaps evolution intended us to, once we've had our kids, you know, there's really no particular reason to go on, one might argue. But, you know, it's not so bad living to be 90, all said and done. But along with that comes the problem that many of us will have dementia. And this is a disorder that comes with the aging brain, though some young people get it as well. And the incidence is quite high. And really, in Australia, currently in 2025, there's over 430,000 people living with dementia of some sort. And it's expected that in 25 years, these numbers are going to double. And so among us here, many of us will maybe suffer from dementia in the years to come. So today, this panel discussion is really all about, you know, look, there are some treatments for dementia. There's no very perfect treatments. But at the Queensland Brain Institute, the Clem Jones Centre for Aging and Dementia, which was set up initially by the Clem Jones Foundation, with a donation from them, is set up to understand dementia, understand what goes on, why does it go wrong, and how can we find cures for it. And that's what the discussion today is about, living with dementia. And, you know, we all recognize many of us will be either living with dementia ourselves or be looking after people with dementia. And it's interesting to have a discussion and think about what is it like to live with dementia. So today's moderator is Professor Roland Sussex. Roland Sussex is a linguist, and I'm sure many of you have heard his dulcet tones on the ABC. And he's a linguist, and he'll be leading the discussion today. Roland.
Thank you, Pankaj. Welcome, everybody. I'm Roly Sussex. I'm a retired but not retiring linguist from the University of Queensland. And this is the third of these Brain Awareness Weeks that I've done. The first one was in 2023 on aging. Last year we did sleep. This year's topic is dementia, is I think by far the most difficult for me in all kinds of ways, which we will discover. The QBI is an absolutely fascinating place. You walk in the door and you can hear the buzz of clever people. It's a marvelous place to be. And this is Brain Awareness Week, and the title of Brain Awareness Week is Living Well with Dementia. We'll come to those terms in a moment. I'm the ringmaster, but hopefully a fairly retiring one, to let the clever people tell you what is going on. The word dementia itself has an unhappy history. It started being used in English in the 16th century, and it meant behavior which was disorderly or undisciplined or inexplicable. And around about the same period, the word demented started being used in the same way. Both of them pejorative, of course. In the 18th, 19th century, eventually we get a medical use of dementia. In 1806, we borrowed the word démence from French. And in 1905, Dr. Alzheimer had a patient with rather unusual symptoms. And he was the person who was able to identify a lot of the properties of the brain and what was happening to it as a result of what came to be called Alzheimer's disease. As Pankaj says, the brain is our most valuable organ. It weighs about 1.4 kilograms. It's about 2% of your body weight. It consumes 20% of your energy. It is not very ecological. But we need it and we must respect it. On the other hand, dementia itself is a disease, and we'll be talking a lot about that today. Now, the word dementia is from Latin, de and mens. It means literally out of your mind. And so it starts with a baggage of things which I'm afraid help a lot of people to talk about it in less than respectful and empathetic terms. It's a bit taboo in many cultures, and I've looked around a number of languages. In Chinese it is shizhi, which means loss of wisdom. In Russian it's slaboumie, which means weak-mindedness. In Turkish it's bunama, which means senility. In Hungarian it is burtalás, which means stupidity. You can see how a description is difficult when the word is carrying so much baggage as well. And worst of all, the word Bedlam, which comes from St. Mary of Bethlehem. Bethlehem gave us Bedlam. It was a hospital in London for the quote-unquote weak of mind. And in the 17th and 18th centuries, people would actually visit there for diversion and amusement, looking at the inmates. We can be cruel, we really can. Parallel to the word dementia, there's about 150 other words, all of which are negative. Words like deranged, crazy, lunatic, insane, mad, unbalanced, unhinged. This is what the dictionary says are synonyms, more or less. And we've got to free ourselves from that kind of burden in order to get a proper view of what's going on. One way to do it is with euphemisms. President Obama signed into law something called Rose's Law in 2010, where mental retardation was officially rebadged intellectual disability. Well, that's a noble intention, but unfortunately, as soon as you create a new word, it acquires the negative things which are hanging around with the old one. The DSM, which is the Diagnostic and Statistical Manual of Mental Disorders of the American Psychiatric Association, has called it a major neurocognitive disorder. Again, big terms, but they have the same problem as dementia itself. A couple of facts which I accumulated, which I want to share with you as the start of this, things which, as a non-dementia specialist, shook me. First of all, dementia is the second most common cause of death in Australia, and the first among women. All right, now that is something really quite shaking. Together with coronary disease, cerebrovascular disease like stroke, COVID, and lung cancer. These are the 5 biggest causes of death in Australia. And among women, dementia is number 1, and in men is number 2. So this is really important. And again, in gender, more women have it than men. For every 1 male that has dementia, 1.4 women will have it. So there is an important imbalance, if you like, in gender terms as well. In age, it affects especially older people, although you can get young onset dementia. And for a lot of people, dementia is out of sight and out of mind. Well, for us today, it is neither out of sight nor out of mind. And what we want to do is to tell you what it is and how it works and what is being done about it. So living well with dementia, not suffering from, these are far too passive and negative. Living well with dementia. The with is now a common way of talking about many kinds of afflictions, diseases, problems, but it means that the disease is separated from the individual, living with. Right? It accompanies you rather being entirely subsumed within you. Living well with dementia. What can you do to live well with it? And living, because there is life with dementia and there's life in dementia, and you'll hear about that from our members of the panel. There will be questions, and on the screen there will be a Q&R code. Point your phone at that, and you will get a little icon on the screen. Tap on that, and a window will come up where you can submit a question, which we will get to in the Q&A later on. You can sign it if you want. You don't have to. It can be anonymous. And the questions which don't get answered will be part of a blog which we'll be talking about later on. So here we go. First of all, introducing the panel, and right at the start I should say that two of the people here are closely associated with the personal experience of dementia, and neither of them has anything to do with the professional staff of the QBI on my right. They will introduce themselves briefly. Jim, you're it.
Yeah, my name's Jim Rogers. I'm a dementia advocate for Dementia Australia. I was diagnosed with early onset Alzheimer's in 2021. So yes, I'm so sorry, I didn't realise that's what we were doing. This is part of it, so I can blame Alzheimer's for everything now.
That's Jim. Dom has a different relationship to dementia. Dom?
Yeah, my name is Dom O'Leary, and I was a very passionate and driven advocate for my father, Noel O'Leary. He was 81 and he had vascular dementia, and he passed away in July 22. I am here to share a story just to tell everybody that you can reach in through dementia and share some stories on how we did that. I still advocate for Dementia Australia because I'm very passionate about inspiring people to reach in and connect when they think they can't anymore, and also empowering people to never give up and keep persisting for the best care for your loved one. When my dad was diagnosed, we were grief-stricken straight away. We went into anticipatory grief, my mum and I. I cared for my dad alongside my mum, who's here in the audience with the beautiful flowers on her hair. And we had grief the whole experience, but we did find, when we reached in, we found moments of joy, and we were able to give him a better quality of life by reaching in.
Wonderful. On my right, your left, we have three members of the staff of the QBI. First, Associate Professor Stephen Zurin.
Yes, hi, I'm Stephen Zurin. I'm a scientist at QBI, and I'll talk about later what I work on, but a very molecular level aspects of how our neurons work and how diseases like Alzheimer's might be caused.
Next is Professor Lizzie Coulson.
Hi everyone. I'm a professor at QBI and also at the School of Biomedical Sciences, who teach a lot of the people who go on into medicine and into research as well. Into neuroscience, and one of the other things I've done for the last 12 years is be on the board of the scientific panel for the Dementia Australia Research Foundation. So I see a lot of the applications that people put in, both for the discovery research and also the psychosocial research.
And finally, Professor Peter Nestor.
Hi, I'm a neurologist by background and a clinician scientist, so my research all involves people who have these diseases. We do cognitive research and imaging, and I run a clinic at the Mater Hospital, and that's where we recruit our research subjects from, so we provide care over there.
Lovely. Okay, now to begin with, and again, I was in the position of, I imagine, a number of people in the audience. I was not quite clear about Alzheimer's and dementia and how many kinds of there are. So Peter is going to start by telling us about what sorts of dementia there are and how to identify them.
Sure. So dementia, as you've just heard, is just a descriptive term for losing mental abilities. So it's not really a diagnosis. Can't hear me. You're kidding. Hello? Yes? Okay. Dementia is just a descriptive term for losing mental abilities that you had before. Okay? Sorry, it's still a problem? No? Okay.
Should be okay.
So it's like saying you've got breathlessness or diarrhea or something like that. It's a symptom. It's not a diagnosis. And then there are diseases that cause people to lose their mental abilities. And so that's where a term like Alzheimer's comes in. The commonest disease that causes dementia is Alzheimer's disease, but it's not the only one. And the other big ones are particularly Lewy body disease, which is related to Parkinson's disease. It's the cognitive part of Parkinson's, if you like, and a group of conditions that are collectively known as frontotemporal dementia. So they're the major degenerative conditions that can lead to dementia. So the point there is, and then there are literally dozens of other rarer and progressively rarer conditions. So this comes up a lot it causes a lot of confusion. I get asked things like, do I have dementia or is it Alzheimer's disease? Well, if you have dementia, it could be due to Alzheimer's. So think of it like breathlessness, and then what's the cause? Is it asthma? Is it heart disease, etc.? And they're the major categories.
Thank you. Now, the other thing that I didn't know about is the structure of the brain itself. I went to your Wikipedia and in 2 paragraphs was intimidated. So I thought we'd do something about that. I've called it The Brain for Dummies, and Lizzie Coulson is going to give us a presentation of what the brain is and how it works.
Great. Can everybody hear me okay? Okay, so here we have a picture of a cauliflower, and your brain is about the same size and weight, as you've heard, of a cauliflower, but unlike a cauliflower, it's quite squishy, and that's why we have our skull around us, and you can see that in the second picture. And that sort of pink blob there is made of cells, and these cells are, a lot of them are neurons or nerve cells of the brain. And there's also another set of cells called glia, and they help support the brain, help support the neurons. And then there's also blood vessels as well that bring the nutrients into the brain and carry away the waste. So in our brain, we have billions of neurons, and they're all shapes and sizes. And there's the schematic at the top there, where you've got the blue part of it, which is what's called the cell body, and then you have an axon or a connection, and then the little tufty bit down the bottom in that first unipolar cell. You can see the schematic, there's different sorts, and then also under that, in the black and white, are actually drawings of a neuroanatomist who traced out the cells many 100 years ago of the different sorts of cells in the brain, and each cell has a different function. Then up the top, you can see that they're organised in different ways, and these are just different slices through the top of the brain, the cortex, which does a lot of the computation that happens in our brain, and they're organised in very particular ways. And just down the bottom is another part of the brain called the hippocampus, and you may have heard about that in press releases and so forth, and this is a particularly important part of the brain for our memory, particularly our short-term memory. That's where we think it gets housed before it transfers into the top part of the brain, into the cortex, where it can then be stored for a long, long time. So what's important is that these neurons are born when we are born, and for the most part, they don't regenerate. So those same neurons in your brain now are the same ones that you were born with. And as we get older, obviously our cells, might have some problems, and so we lose our cells in the brain. So then I want to tell you a bit more about the neurons. Think of them like an old-style telephone. So the handset is the tufty bits, and they make the connections. They are the receiver and the microphone for sending out the information and talking to other neurons in the brain. And at that point, we have the synapse, and that's where what's called a neurotransmitter, or the little signals of the microphone, get released and transported across a tiny little gap to the next cell to communicate with the other cells. Much like, again, an old-style telephone exchange, where there are lots of wires all over the place, these connections are not just one-to-one. So we have 1 billion neurons, but each neuron may well have thousands or hundreds of thousands or trillions of connections with the other neurons in your brain. So down at the bottom there, that funny squiggle, is actually a worm called C. elegans, and this is a worm that Stephen hopefully will talk to you a little bit about, or at least that's what he works on. And we know in this worm that there are 300 nerve cells, and we have mapped every connection between those nerve cells in the worm. But there's only 300 of them, and that's quite a complicated diagram. So imagine all of those connections in a human. There are lots. And I don't know what happens next. I think we'll get another picture. No. So anyway, in humans we can map We don't know exactly where everybody's neurons are and where they connect to, but it's those connections that underpin the function. So I imagine that many of you will have come here because you're interested in the brain, and you might be interested in what is genius or what is schizophrenia or what is dementia. But we have, before we can really answer those questions, we have other things that we need to know as well. How do we learn, forget, think, dream, love, have internal conversations with ourselves? How do we remember the sights and sounds and smells from 5 or 50 years ago? They are just fundamental questions about our brain. And if we don't know how we learn or forget, how can we even begin to understand what dementia might be, and therefore how diseases like Alzheimer's might form? And so at the Brain Institute and neurosciences around the world, we're trying to understand what neurotransmission is, how different neurotransmitters act together, what keeps neurons alive for 100 years, how is the waste carried out and nutrients carried in, and so forth. You can read, because we still don't know the answers to those questions. Next slide. So the key features, and you saw a little bit on the slide with Peter, the coloured pictures for Alzheimer's disease. So this is a cross-section cutting through the brain in a diagram, and we have a healthy brain on one side and an Alzheimer's brain on the other. And you can see the Alzheimer's diagram, it's smaller and shrunken, and there are some bits that are actually missing. And you can see the little, hopefully you can see there's a little curly bit that's the hippocampus, and that area is missing in this schematic of somebody who has died with Alzheimer's disease. So it's not surprising if that part of the brain is missing that they can't have any short-term memory anymore. The neurodegeneration underpins the cognitive impairment. So that's something we know. And in Alzheimer's, the areas of the brain that degenerate correlate with the symptoms of someone with Alzheimer's disease. But that would be different for somebody with frontotemporal dementia, for example. In Alzheimer's disease, there are brain regions that degenerate, and one of those brain regions is called cholinergic basal forebrain, and you can see the little dots are the cells in the brain, and there are less of them in Alzheimer's disease, and they degenerate in both Alzheimer's and in the Lewy body or Parkinson's disease style dementia. And they are one of the targets of the current treatments for Alzheimer's disease that have been around for the last 10 or so years. And then there is also the pathology. So this is when we look deep into the brain at the cell level. We have both plaques and tangles, the amyloid plaques and the tangles, and you may have heard, again, those things on the news. And a recent treatment that was approved by the FDA is targeting the amyloid plaques, but it's a little unclear. Peter can talk a little bit more about whether it's effective or not. So I think that's it for Brain for Dummies.
That was magisterial, putting together all of that into one beautiful, coherent picture. All right, we now move on to the presentations by the 5 members of the panel to give you different perspectives on dementia and how it affects their lives and how they work on it from the point of view of their science. We start with Jim and living with dementia.
Disease of the brain that strips you of the things you hold so dear, which are your memories, people that you love, and the thought of me not recognising Ty or my kids or my grandkids like that, I just can't even think about that, because that is all, that's everything, isn't it, really? Good evening, everybody. Firstly, I'd just like to thank the QBI for inviting me to take part in this really important evening. That clip that you just saw, that was myself being interviewed by The Imperfects, which is Australia's top podcast for mental health. And they invited me to Melbourne to go and do that interview. So it's been a long road to get to that point where that interview happened. And so I'll take you back a little bit. I was working as a property developer in a property coaching company in Sydney, and had a job with quite a lot of pressure and a big team of people and very chaotic circumstances. And I was experiencing a lot of fogginess and cloudy information. It wasn't correct to me. I was meeting people, having conversations, and then not remembering the conversation, which was really alarming, because to continue doing that sort of work where you're making big decisions, those get them wrong, and it can really have a big effect on the entire project. So even colleagues were pointing out to me that I'd met people face to face, and then the next time I met them, I had no clue that I'd ever met them before. So it was all a little bit odd, and I just was getting a bit overwhelmed by it. And every time I went to my GP, I would mention it. So if I went for a routine appointment, I'd have to say, I think I'm struggling with these things happening to me. And he used to say, okay, let's do this little test and remember this and do that. And to be fair, I always was fine. So I literally put it out of my mind. And then sometime later, I went to a cardiologist appointment. And my cardiologist was amazing and very smart, switched on lady. And I was sat there in the room, and my phone was ringing and ringing. I was busy, and so people were trying to get hold of me. And she kept giving me a little bit of a look as if to say, okay, I think you're being a bit rude now, put your phone on silent. And I could not do it. I just could not work out, how do I put this phone onto silent? It was so embarrassing and so stupid, I just couldn't work it out. So I eventually said to her, could you put the phone on silent for me? And she was onto it, and she watched me, and she said, no, just do it, take your time. I tried, and I could not do the most simple task, putting your phone on silent. So she said, are you experiencing some weird things like this happening? And I was, yes, absolutely, this is exactly what's been happening to me. So from that point, she referred me to a memory clinic, and the memory clinic started to do deep testing. They did cognitive testing, how the information goes into your brain and how your brain perceives that information. I'd be here all day if I explained it in detail, but it is quite intense, and some of it I sailed through, not a problem. Some of it I failed miserably in. Enough to alarm them into telling me I'd got cognitive issues, and they wanted to do more investigation. So from that point, my neurologist sent me for a brain scan, and I had various things done, and then eventually I went for a PET scan, and they use a dye, and they send you in, and you have all of that done. So eventually at the meeting with the neurologist, she explained to me that unfortunately from the PET scan, they could see that it was early onset Alzheimer's, which I think we call younger onset Alzheimer's now, which was just so shocking to me. I couldn't even believe that was something I was going to be facing. I literally shut down from that point and found it too hard to absorb, and I went away and started to look into what Alzheimer's was. I knew nobody with it, I knew no information about it, but I soon discovered it was a very depressing outlook, especially in younger onset, which it can often be more rapid. I started to watch films that involved that, and it was very alarming, so I was quite depressed for a while. And then eventually my neurologist referred me to Dementia Australia, and as soon as I connected with them, they took me under their wing, and I met the lovely Kylie, who is here, who helped me with organising a course, which is Living Well with Dementia, which I can't recommend enough, and was brilliant. It teaches you all about doing your will, very confronting things, getting your life in order, your medical, I can't even remember the terminology now, but all of those things, it teaches you about what's going to happen and how you can change your diet and exercise and and be positive, and etc. A lot of those things. Anyway, we got on very well, and so they asked me if I would like to do some advocacy work for them. And then from that, they created Australia's first podcast called Hold the Moment. So you can all go and listen to that. And I was very excited to be asked to be a co-host. And then I couldn't believe it when they said it's going to be with Hamish MacDonald, who's the renowned journalist and presenter of the project. So together we've done this podcast, which is just close to 100,000 downloads. So it's been huge for dementia, and it's great for people who are touched by it or suffering with it. And because of that, Hamish, who is famous, was asked to do the podcast interview for The Imperfects, which he did, and while he was there, he brought up the topic of dementia and recommended to them to interview me because I was doing the podcast with him and I'd been diagnosed with Alzheimer's. So they flew me to Melbourne and I did that interview, which is on YouTube if anybody's interested to watch it, on The Imperfects. And so something so awful, which has been devastating to me, turned into something which was quite good. And I tried to remain positive, I tried to stay upbeat. And I think everybody's experience with dementia is different. So if you can do things that are good and keep you motivated and happy, then that's the way to go. So I hope that was an insight into my journey.
Thank you, Jim, and that definitely is one way of living well with dementia. Dom's experience was very different.
As a carer for my dad, I'm very passionate about two things, sharing these two things. One is busting the myths that come along with dementia. Number two is boosting connection. I think it's really important to bust the myth of dementia that people say it's just normal aging. Dementia is a brain disease and it is fatal. I think if we think of it as just aging, we become more passive in our approach to care, and our loved ones don't get the correct care that they need, and we don't reach in because we're just passive in our approach. You often hear people say, what's the point of visiting someone in an aged care home because they won't know if I visited? Or what's the point? It's so sad, they have no quality of life. But to me, quality of life is reaching into your loved one and connecting and making their life better. Boosting connection. How do we boost connection? So for me, boosting connection was celebrating who they are with them. So how we do that is we reach in and we think about the things that our loved ones loved or do love, and we do those things with them and celebrate those things with them. One of my favourite quotes, that's my beautiful dad there on the left. He was a very funny, charming man, actually. He used to answer the phone with a dry sort of tone, and he used to say, "City Morgue." I think it was for telemarketers, but yeah, he had a great sense of humour. One of my favourite quotes, and I think it sums it up, is to love a person is to learn the song in their heart and sing it to them when they've forgotten. So Mum and I used this and we sung, we danced for Dad. We used his favourite sayings. He loved Arnold Schwarzenegger, so we used to say, instead of goodbye, because it was so hard to leave the aged care home at the end of the day, he ended up going into aged care because it became dangerous for him to live at home, but we would say, "I'll be back." And he would giggle, and that would sort of dissipate the tension of having to leave him there. I first started bringing up a pile of photos for Dad, and I thought, I'll show him this photo of his old car. I knew nothing about this car. So I showed him this photo and he looked at it, and then he sort of shut his eyes and went to sleep. And at this point, Dad was sleeping a lot and not really engaging with conversation. He was more non-verbal. I started talking about the car, thinking, did it go like stink? Did it have a nickname? I wonder. And then all of a sudden, out of nowhere, my dad, who hadn't spoken for 2 weeks, said, "1500" in an Italian accent, and I never heard him speak Italian before. And I Googled it, and it meant Fiat 1500. And I was just amazed that I had learnt something new about my dad through connecting through these photos. So I decided to make a life book for him, and I collected lots of photos from Mum, wrote stories about our travels together. I had his siblings write short stories about when he was young, and we just compiled it all. I'd bring up a page every time I visited, so he had something to look forward to. But I really did it because I knew as he was declining, and it was such a heartbreaking journey and he wasn't speaking, that I would have hard days and I wouldn't be able to keep up that constant talking. So I could read to him from it, and every time we did read to him, he would react or smile or say something. So I think that tool was just so powerful. I highly recommend people doing it. The other story I really want to tell is non-verbal communicating with your loved one. You'll see up the top there's a picture called the running hug. One particular day, Dad was not reacting at all, not even smiling. I did everything I could to get him to engage, and he wasn't really engaging, and I gave him a hug and he just didn't react, and I just felt so sad, and I thought, no, I can't leave like this. We got to do better. So I told him, okay, Dad, this is going to be an even better hug because that one sucked. So this hug is starting from over the other side of the room, and it's going to be huge. So I started running in slow-mo towards him, and I was describing what I was doing. So here it comes. Here comes the biggest hug you're ever going to get. And as I got closer and hugged him, he just burst out laughing. And he giggled so much, and Mum took that photo. She laughed so much, and we were just laughing. And I thought, this is like such a meaningful moment of connection that we wouldn't have had if I hadn't have tried harder to reach in. So yeah, I just want to share these stories and empower people to just celebrate who your loved one is with them, learn about things they love. I'm a peer support person with Dementia Australia, and I have a peer at the moment who wasn't connecting with her mum, and they were arguing, and I said to her, what is the problem? You should be going with where she is now. Don't disagree with her, just meet her where she is now. So agree with everything she says, and then find things she loves and do those with her and celebrate her with her. And she ended up bringing in some beautiful Beatrix Potter dolls that her mum used to love, and reading her stories, and she's finding connection, and she said it's so much easier to visit her mum now. So yeah, I hope these stories help other people.
Don, thank you. What a lovely life-affirming way of caring for someone. Beautiful. We now move, I'm, if you like, the membrane between the life experience and the scientific experience. We now move to the scientific and medical part of dementia. And our first speaker is Associate Professor Stephen Juren, who's going to be talking about cells and mitochondria, something else that terrifies me. Stephen.
Thank you, Rolly. It's a bit of a change of pace going from those really human stories down to now a molecular level and a very scientific level, but I'll try and keep it going well. As we've heard about today, the brain obviously is the important organ in our bodies that allows us to think, but is composed of around 100 billion neurons, which Lizzie introduced beautifully just before. But we're going to go down even deeper now and inside the neuron and look down at that real molecular scale. And within each of our neurons, we have these little structures called mitochondria. You can see them there up in the blue. And there's about 1,000 or 100 to 1,000 of those little mitochondria in each one of our neurons. And these are the power factories of our cells, as well as our neurons. As Rolly mentioned at the beginning, our brain uses 20% of our energy, but it is only 2% of our weight. It's a very energetically demanding organ. The neurons are constantly firing electrical signals, and it takes a lot of energy to do that. The mitochondria produce this energy. So every time we eat food, we actually burn that food with the oxygen we're breathing inside of these mitochondria to generate a chemical energy called ATP, which is the life force of everything living, plants and animals. Plants and animals all have mitochondria. And what's special about mitochondria is they have their own DNA, which exists as a small, round, circular DNA molecule within each of our mitochondria. And this is separate to our nuclear DNA, which is in our nucleus. And we only inherit this mitochondrial DNA from our mother's side. Our fathers don't pass down mitochondria in the sperm. And this DNA that's inside the mitochondria is basically an ancient remnant of when mitochondria used to be a separate species altogether. They used to be a bacteria. This is how the theory goes. Around 2 billion years ago, we basically engulfed these bacteria into our cells, and they became endosymbionts. And today, they still carry their own DNA. And they actually do look like a bacteria. The important thing with this DNA, it's the blueprint for for the construction and the function of these mitochondria. But because these mitochondria are like power factories, they generate these toxic chemicals, these byproducts, called superoxide and reactive oxygen species and free radicals. You may have heard of them. That's because when we breathe in oxygen, not all that oxygen gets consumed properly to burn the food, and some of it actually gets converted into superoxide. And that DNA inside the mitochondria is therefore exposed to these toxic chemicals, which can lead to mutations in this DNA. And this happens as we're breathing here today. It happens as we age. It gets worse and worse, so that eventually we accumulate more and more mutations. And this is perhaps one contributory factor towards why our neurons degenerate during aging and leads to certain diseases like Alzheimer's disease. If we can maybe go to the next slide, this is what the actual mitochondria look like. This is a human cell, and all those little jiggling things around in purple, they're the mitochondria. So you can see from this sort of video, they do look like bacteria, and they're full of energy, and they're critical. And this is what we study in my lab. I won't talk about worms today, that Lizzie brought up before, but we do study them using this simple animal model called C. elegans, a very simple animal, and it allows us to get in there really deep. They're actually transparent. We can see through them. We can see the mitochondria and study what goes wrong when that mitochondrial DNA becomes mutated, and we're actually trying to understand how we can repair that DNA to prolong the energy supply so that our neurons can survive better as we age. And so the mitochondrial theory of aging and of age-related diseases is just one potential component, and there's many other theories about proteins within our cells and amyloid beta and et cetera. But it's something that we do at QBI is we try and tackle all these diseases from many different angles and different approaches.
Fantastic. I shall respect my mitochondria from now on. Okay, that is at the very deep level inside the cell. We now pull back a little bit, zoom out, and Lizzie Coulson is going to tell us about lifestyle things and sleep apnea and how they relate to dementia.
Great. I can already see on some of the questions that we're getting a preview of that there's lots of questions about things like exercise and food and so forth. And please correct me, Peter, if I'm wrong, that I think living well with dementia and trying to stave off diseases like Alzheimer's disease, all of the things that you hear about in terms of green leafy vegetables and exercise and red wine and dark chocolate and sex and, you know, add whatever you want in there. These are about living well, and certainly we know that a better cardiovascular system is something that can, you know, slow down or stave off vascular dementia. But at the moment, there is nothing that I know about that is actually going to stop you getting dementia if the genes and the environment that you've grown up with is leading you down that path. But it will help you to live well with what's coming. So science is, or neuroscience is like a big jigsaw puzzle, and we sort of maybe have an idea that what the pieces are around the edges maybe, but there are lots of pieces that we don't know what they are yet, and we don't even know what shape they are and what the big picture looks like. And so as scientists, we come up with what we think the picture should look like, which is what we call a hypothesis, and then we investigate that. And I'm just going to take you through our work over, some of our work over the last few years. So we know that middle-aged people, about 15% of them, have obstructive sleep apnea. So this isn't snoring, this is actually when you stop breathing during your sleep. And it's actually about 50% of our elderly. And people with obstructive sleep apnea have 3 times the risk of developing dementia, and in particular Alzheimer's disease. So we tried to understand why this association might be. Can we understand the cause of this correlation? And we knew that cognitive decline correlated with the cholinergic neuron death, the cells that I told you about earlier that die. And so we took mice and we gave them obstructive sleep apnea, essentially, and we studied what happened to their memory and what happened to the pathology in these animals. And we found in these animals that it was the hypoxia, or the lack of oxygen, that happened in these mice, rather than the poor sleep, that caused increased problems with the memory of the mice and also increased pathology, the amyloid plaques that accumulated in these mice. And so we think that that's what the risk is. And then we gave our mice CPAP. Not quite. A CPAP is continuous positive airway pressure, so it's the elephant mask that people get when you're diagnosed with sleep apnea. And so we did that with our mice, essentially, and we found that this stopped these neurons from dying, and so potentially can stop the risk. Now, we know that's just in mice, but we also found that 40% of newly diagnosed people in middle age, so 55 and older, have cognitive impairment. And some of those people probably, we had to refer them on to the memory clinic because they had sufficient impairment in our study that would have been picked up as dementia. We also know that 50% of people who are diagnosed with obstructive sleep apnea and for which CPAP could help don't use their machines. They're not good at it, they don't like it, which is fair enough. It's not particularly nice to use. And they might prefer, for example, to sleep in a different bedroom so that their bed partner doesn't have to cope with the noise and the discomfort or the snoring. And so now we're at the point of who should we target in this sleep apnea population and how should we do that? So one thing is if we know who is going to develop Alzheimer's disease, in the vast majority of people, so a lot of people have sleep apnea and don't get dementia, but if we knew which ones did, well, we could target them with therapies or just with CPAP compliance. And so we're trying to develop new drugs that stop the cell death happening, but we're also trying to understand the volatility to work out who with sleep apnea is most likely to get Alzheimer's disease. So just if you look here, you can see the little green line wiggling around. If you imagine that is sort of your breathing or oxygen level, and you can see various variations or volatilities of people with sleep apnea, and some of them have lots of yellow bits and some of them don't, and some of them have quite a lot of time above the line and others don't. And so we're looking at how people breathe, mimicking that in our mice to see what causes the cholinergic degeneration, and then hopefully flipping that back across to people with sleep apnea who we can then follow and see who has developed dementia, and if we treat them with CPAP, whether that's only actually going to increase or protect their memory, whether it's they've had sleep apnea for so long that it's permanent damage that we can't fix with CPAP, or whether we can actually have a public health campaign to use your CPAP. So we'll leave it there.
Thank you, Lizzie. We now move on, or before I do that, if you look at the bottom corner of the screen when we get to Peter's slides, that is where you can get the image which will activate Slido, which will communicate your questions to our team at the back, and they will turn up later on for the team. So if you'd like to put questions, look for the little QR code at the bottom there, and that's how you can connect your phone to the software which will collect it. All right, thank you to Lizzie. We now move to a slightly different perspective, particularly that of a clinician, someone who sees people in a hospital or consulting rooms with issues of dementia. And our expert here is Professor Peter Nestor.
Thanks. Thank you very much. I made this slide because I thought this might be of interest to people. This is actually part of our research, but and what you're looking at here are statistical maps of the brain. So when you get assessed, if you're suspected of having something, you get sent off for brain scans. And in certain circumstances, the brain scans can be quite helpful, but a lot of the time they're not very helpful, such as in the beginnings of Alzheimer's disease. And the reason they aren't very helpful is kind of makes sense if you think about it. You're dealing with a disease that's coming on in a sort of a gradual kind of way. It's not like a stroke where suddenly a whole piece of your brain just got damaged from a blocked blood vessel. It's just coming on in a very insidious kind of way, and you're losing connections between brain cells and you're losing cells. But also, we kind of lose cells as part of normal ageing as well. Our brain shrinks as we age. So given that brains shrink as we age, and it's just slightly more shrinkage if you're developing something like Alzheimer's disease, you can see that looking at a scan and eyeballing it and saying, oh, there's a significant amount of shrinkage there doesn't really make a lot of sense when you think about it. I'm sure when you've got advanced disease there'll be obvious changes, but people don't go to their doctor when they've got advanced disease, they go at the start of the illness. So one of the things we're working on is trying to make that decision about whether there's brain shrinkage more objective. And so what you're looking at there is statistical maps where we're looking at the cortex of the brain, the outer part, the grey matter, and we're turning areas where there is abnormal thinning of the cortex into a statistical map of colour coding. And so the more red it is, the more thinned it is. And and those maps are generated in comparison to the healthy population. So with our research, we also have older people who've got nothing wrong with them coming and seeing us all the time as well, who very kindly help out with our research to create a sort of a normative range. So that's the background to these statistical maps. But the reason I wanted to show them to you, I've just used 4 examples there of different diseases that cause dementia to show you that the brain is affected differently according to different types of dementia. So if you look at the one on your left, which is the one that says Alzheimer's, and the top is looking at the inside, the mesial surface of the hemisphere, and you can see that there's a blob there that is very thin. That's called the precuneus and posterior cingulate cortex, and that's really a key area at the start of Alzheimer's disease, that area seems to degenerate first. And then the image below it shows looking at the outer surface, and you can see that there is and to your right is the front of the brain, and to your left is the back of the brain in that lower panel there. You can see there's more red stuff towards the back of the brain. Now, if you then move along to the middle one, which is called progressive supranuclear palsy, so this is in the frontotemporal lobar degeneration family, and this is a disease that also affects motor function and balance. And you can see here that now it's right in the middle of the brain at the top. That's the back of the frontal lobe that's affected. And then to the right of that we have this condition, behavioural variant frontotemporal dementia. And now it's the frontal lobe is where all the red stuff is, sort of if you're almost the mirror image of Alzheimer's disease, the front part of the brain degenerating rather than the back part of the brain. And then we have Lewy body at the bottom there, and there isn't any thinning at all, but it affects the function of the brain. And so if I showed you functional scans with positron emission tomography, it would be a much different picture. Anyway, the point of all this is that different parts of the cortex are affected in these different diseases, and this happens in quite a stereotyped way. So that pattern that we see in Alzheimer's, where it's the back part of the brain and particularly the inside part of the cortex, you see that in person after person after person, and so on with the others. And this, I think, is a really fundamental point that we need to kind of come to grips with if we're going to really get a proper mechanistic understanding of what's causing these diseases, is that they don't affect the brain randomly. It's not just that, in this person it started here and that person it started there. Different diseases have predilections for different parts of the brain, and we see that repeating itself over and over again. And then moving to the symptoms, the symptoms are dictated by which bits of the brain are degenerating. And so, for instance, in Alzheimer's, everyone knows of it as a language condition, but as a memory condition. But as it progresses, it affects language and your visual or perceptual abilities, the way your brain handles visual information. And no surprise, the back part of the brain, for instance, is important for that type of supporting those kinds of functions. At the other end of the spectrum, on the right, the frontotemporal dementia, the frontal lobe is more about decision making and planning and problem solving and so on, and also to a large degree our personalities. And so whereas Alzheimer's might start with memory trouble, frontotemporal dementia typically will begin with changes in personality. And that makes that one a very tricky one to diagnose, and very easy to misdiagnose, because it blurs into sort of psychiatric illnesses. So that's a little bit about these different diseases, and why is it that Alzheimer's affects that bit of the brain and frontotemporal that bit of the brain, and Lewy body doesn't cause it to thin, but the cortex to thin, but it causes it to not work properly. We don't have the answer to any of that, and I think that that's something that we absolutely have to come to grips with if we're going to sort of move to a sort of a next stage of enlightenment, if you like. I thought I might just, a quick couple of comments just on a few other things. We hear the words young onset all the time, and later onset and so on. And I thought it would be interesting just to highlight that what's the definition of young onset versus not young onset? Well, it's an arbitrary cut-off at age 65. So when you say I've got young onset Alzheimer's disease and I'm 64, and someone else, I've got old onset Alzheimer's, I'm 66. So they got two different diseases. No, biologically they have exactly the same thing. And this comes back to a bit what Rolly said right at the introduction. This has its background in sort of sociology rather than in biology. The reason that 65 is the magic figure for young onset versus late onset is just that retiring age was 65. And the idea was that if you started losing your mental abilities after you retired, that was normal. So for instance, when Alzheimer discovered his patient, he didn't think he'd discovered dementia. Ancient Egyptians knew about dementia. What struck him was that his patient was very young. She was only in her 50s. And that wasn't supposed to happen. And then the other reason why it's called Alzheimer's disease is that around the time that Alzheimer did that, a new stain had just been invented. So when these pictures you've been seeing of the slices of the brain, if you just cut the brain and looked at it under a microscope, you wouldn't see anything. But you apply stains to it to show up the different cell types and so on. And a new stain had just been developed around that time of Alzheimer, which enable them to see the pathology of the what was subsequently shown to be amyloid plaques and so on many years later. And so that's why it dates to Alzheimer's. It wasn't that he was the first person to notice that people get dementia. That's probably enough for now.
Thank you, Peter. This is fascinating stuff, and that's why we need people doing advanced and very delicate science to try and find out what's going on in our brains. We now come to Q&A, and there's the QR code if you haven't grabbed it already. We have a number of questions on the screen here, and I'm going to ask the members of the panel if they'd like to address one or more of these. I think we might start with this one, an anonymous question from the audience. My mother has Alzheimer's disease. Does that mean I'll get it?
Yeah, that's an interesting one. You might, or you might not. There are rare forms of Alzheimer's disease where it is due to an inherited gene. This is less than 1% of people with Alzheimer's, and it causes Alzheimer's at a very young age, people in their 30s and 40s. the more sort of standard type, if you like, where people get it in later life, there's a bit of a hereditary dimension to it in that there are genes that are risk factors. So someone who's got a parent or relatives, ancestors who developed Alzheimer's disease, perhaps is a bit more likely to get it than someone who has no family history. But there's another dimension to this, which is that these diseases are a function of ageing. And so this is, I think I told you this the other day, this is an amusing thing that happens sometimes, that I see people and they, I'm cooked, everyone in my family's got dementia. So when am I going to get it? And, well, tell us about your family. Well, there was Gladys, she was 98, and then there was Stan, he was 97. So sometimes what's actually running in your family is that you've got longevity. And so so families where, well, we've got no one in our family, but we're all dead by 75. So it's a complex equation, but in summary, no, it doesn't mean you're going to necessarily get dementia because your mum had it.
Anyone else?
Can I just make a quick comment on that?
Yeah.
A friend of mine back in the UK who actually passed away at 54 from young onset Alzheimer's, had the familiar line, very strong familiar line. And her 3 children, when they became adults, the 2 boys, a girl and 2 boys, the 2 boys did not want to know at all, because you can go and have the gene test, and they just did not want to know. But the daughter really did want to know. So she went and had the test. And fortunately, she hadn't got it. So, everybody was overjoyed with it because their whole family line has had some part of it. And so it encouraged the two lads to go and do theirs. And they did, and they both have it. And they're 27 and 29, and they're now running marathons to raise money and stuff for Alzheimer's UK. But they've got a hell of a journey in front of them. But that familiar line in some families is really strong.
Wow. Dom, here's one for you. What are the ways to care for a carer of a dementia person?
That's an easy one. I would contact Dementia Australia straight away. That's what I did. And I received there's a helpline you can call. It's 24 hours. But I was able to receive some counselling. So I had a counsellor that understood the disease, and I could talk to them about Dad's progression and about my plans and how I was still researching it at the same time, but I could just run things by them, and they were a great listening ear, because often friends don't understand what the disease is, and they really don't want to listen because there's a wall that we put up. So there's 2 walls. I call the wall, the first one, of fear, that people don't want to talk about it, or they're scared to talk about it or acknowledge it, or they're scared to deal with it. And then there's the wall that is dementia, where we have to break through that wall, where our loved ones can't reach out and connect with us because they've lost their social skills and they've lost the ability. So we have to break down both of these walls. And I just think having the support through an organisation like Dementia Australia, who understand what the disease is, they can point you in the direction of short courses. They can just be a listening ear. That's what you need. The peer support program that I'm a part of is just amazing, because you'll be matched with a peer that has similar caring circumstances such as yourself, and you'll get to speak with them, and they'll just get to listen and empower you to look after your loved one, say yes, fight for that in the aged care home, that's not right, and inspire you to reach in and connect. So definitely Dementia Australia would be my go-to.
Where do we go next? How effective are drug treatments for Alzheimer's?
Sure, I can. So the drugs that we have available at the moment are what are called symptomatic treatments, meaning that they don't change the course of the disease particularly, but they can help symptoms. If you have the symptoms that those drugs help, they can be fantastic, actually. So the question is, well, what do they help? There's 2 main types of drugs that are available. One is these things called cholinesterase inhibitors that boost acetylcholine levels, what Lizzie's been talking about. Some people develop delusional beliefs and hallucinations and things like that. Those drugs are really good for that. Lewy body disease is the disease that really has a lot of problems with hallucination, and you can generally switch the hallucinations off with those drugs, at least for a time. As things progress, it'll start to re-emerge. So in the right circumstance, if the symptoms are right, then you can have quite a big impact. Unfortunately, though, they got a bit of a bad name because they got translated into just treatments for dementia or treatments for Alzheimer's, regardless of what the symptoms were. And they're leading people to think, well, took those drugs, they didn't seem to do anything or whatever. But the problem is that they're not pan-Alzheimer drugs or whatever. They're targeting certain symptoms. So if you take someone who's just at the start of the illness and they're just a bit forgetful, they've got no behavioural problems or whatever, well then those drugs don't really do anything at all. The other class is what people really want, though, are things that stop the disease or change the course of it or slow it down or whatever. And that's unfortunately the bit that's been very disappointing so far. And even these new ones that have been hyped a lot in the press in the last 12 or 24 months that are aimed at dissolving amyloid plaques out of the brain, which is one of the features in the brain of Alzheimer's disease. And they actually do that. They work very well at dissolving the amyloid out of the brain. Unfortunately, though, it doesn't stop the disease. It keeps continuing, and which tells us something that I'll finish off at the end of the night on that point. But it really underpins or drives home the point that we need to be looking at other things and not just perseverating on the idea of getting rid of amyloid and it'll all be okay, because those studies have shown that that's not the case.
We're going to run out of time. I should say that the questions that haven't been answered will be picked up in the blog, and you'll be able to get at that on the website of QBI, so that the questions won't have vanished and they will be addressed in due course. I think a symptom of Lewy body seems to be sleep apnea. Cause or effect? Lizzie?
Probably Peter's better to answer this one as well. I think the real answer is we don't know, and maybe the sleep apnea is unrelated to the Lewy body, but we know people with Lewy body do have different sleep problems, and so maybe that's exacerbating the issues. Peter?
Yeah, I wonder if you're thinking about something called REM sleep behaviour disorder, actually, with the person who posed that question. So there's this weird phenomenon that you get with Lewy body disease in sleep. REM sleep, you've probably heard, rapid eye movement sleep. That's when you're dreaming. And when we dream, our bodies become paralysed and our eyes flick around looking at what we're dreaming at. That paralysis that happens during dreaming gets lost and you start acting out your dreams. And so the bed partner gets a black eye or something because the person's asleep and dreaming that they're fighting off the tiger or whatever. And that's the pathology underneath Lewy body is this stuff called synuclein. And for some reason, diseases that cause synuclein to deposit in the brain have this strange effect of causing this thing called REM sleep behaviour disorder, which we use from a diagnostic point of view. When you hear that story, it puts you down the Lewy body direction.
Stephen, the last question on the screen is one which interests me. Are there any improvements on the horizon to make screening process easier, to hopefully allow an easier diagnosis? You want to have a go at that?
I'm really not the right person to
do
without any kind of medical. Yeah. I can answer basic science questions about what's happening at the molecular level, but not with any patient care, unfortunately.
Okay.
I think one of the even though we've had maybe a disappointing last 20 years where we thought maybe the getting rid of the amyloid out of the brain might do something, even though we haven't got the miracle cure we were hoping for, what we have discovered through that process and putting the jigsaw pieces together is a lot more about what is happening in the brains of people living with dementia and before they get a disease, in terms of their brains and the pathology and so forth. So one of the things is, though, that the way our society works is that we don't want to screen people unless we can treat them well. And so while there are lots of things that are being used for screening for research purposes, whether they're new cognitive tests or new ways to image the brain, either looking for amyloid or looking for changes in the way the brain functions, or looking at the cholinergic system or the energy metabolism. These are things that we can use, and but we're not using yet. But as soon as maybe we have a good treatment, then that might be the way that everybody can come in at age 65 or when they retire or something like that and be screened to see whether the available treatment is something that's going to work for them. Or be put on a list for, okay, we'll monitor you and go down that way. At the moment, those screening tools are very expensive, so they're unlikely also to get put into the public arena, and so there are a lot of people working for other ways to screen people, blood tests, for example, but these are very hard to know. The changes that happen in your blood, are they actually representative of something that's happening in your brain and something that might happen in the next 10 years or 5 years?
I think we'd better start wrapping this up because, as you see, this is a very big and complicated area. But one of the most important things that's come out is that, in fact, dementia is a disease. And humans are actually rather good at dealing with disease when they get motivated enough. We have actually entirely eradicated smallpox, which killed, I think, 500 million people during the course of its ravages over the centuries. We've almost got rid of polio. We've made huge progress with cancer over the last 50 years. And so if dementia is in fact a disease and is recognised as such, then there is something which my colleagues are working on and where we can hope for an outcome. I'd like to ask the panel, in 1 minute each, what they think the most important next step is from their point on the map of dementia. Jim, you're first.
Why me? You keep throwing it to me first.
We're going in a circular fashion.
Sorry, can you repeat the question again?
Yeah. What would be the next most important step in work on dementia from your point of view?
I think for all of us, it's looking for the potential for a cure, and it just is so complicated, that brain, how we find that answer. I mean, for me, I watch and read a lot about what the possibilities are, but I do understand it's very complex. And listening to the experts who are so knowledgeable, it's mind-boggling. I don't envy you guys at all. It must be so hard.
Dom?
I think at the same time we are looking for a cure scientifically, we need to be creating more awareness that the social and psychological impacts dementia has on people, and by sharing and talking about dementia openly, coming to talks like this, donating to the Brain Institute or Dementia Australia, researching and just listening and sharing stories, we are creating more awareness, and by creating more awareness before we've actually cured it, we can still help people that have the disease at the moment. So I think that's really important.
Great. Stephen, as a scientist, what is the next most important thing?
Well, for me, it's really understanding how our brains work at every level. I mean, for me, I use the analogy of fixing a car. It's really hard to fix a car if you don't know how the engine works. You don't know what the tubes are doing here and what it does that. You just end up guessing and doing trial and error. And so, for me, working out what these little molecules are doing, because each of our cells is almost a universe within itself, yet we don't know what all of those little molecules are doing and what they normally do to function correctly, let alone when things go wrong. And I always think that positively, I'm quite optimistic that it will be solved. Like, this is just a physical problem. This is within our grasp. It's not a metaphysical question or something spiritual, which is we can't answer. This is something that's very physical. We're beginning to develop the technology in the last 50 or so years to really understand what all those molecules are doing and what happens when they go wrong. So for me, it's a matter of time before there's a treatment or a cure.
There's a good word. It's a matter of time. Lizzie?
Oh, I agree with Stephen. I think it's more research. We have to understand the fundamentals, but we have to put the pieces together as well. So it's lots of people doing lots of different research and then having the insight to go, oh, I'm working on the cholinergic neurons, but hang on a second, they're some of the biggest neurons in the brain. Maybe they're dying because of the energy metabolism, so maybe I should work with Stephen. So it's putting the pieces together and looking to see what other people are discovering to help all of it go forward and to see the bigger picture.
Lovely. Peter?
Yeah, I think that it's admitting that we still don't know what causes this. So we observe abnormalities, pathology in the brain, and for most of my career, focus has been on removing that pathology from the brain and hoping to get better. And that's was an entirely appropriate thing to do. There's something abnormal in the brain, get rid of it and perhaps it'll stop. But that, considering that hasn't worked so far, I think we need to start really embracing the complexity of it and not succumbing to soundbite culture of just get rid of this and everything will be okay. And looking at understanding why certain parts of the brain degenerate. It's not, as I said before, it's not just everywhere. It's certain areas in a stereotypical kind of fashion. And understanding what makes those cells vulnerable and what things could be done to make them more resilient, which is why things like understanding how mitochondrial function works or how synapses talk to each other and things like that. So having a much more kind of embracing of the complexity rather than trying to dumb it down and just say, oh, you just have to eat cabbage and you'll be fine type approach.
I'll go with that, particularly given my view of cabbage. Okay then, look, thank you, panel. This is a disease, and as a disease, as Stephen said, it's a matter of time. And clever people doing clever things to try and understand how to tackle it and what to do with it and about it. Something that Dom said was important, I think, that we need better and more open communication. Remember, out of sight, out of mind is very much the way we've tended to treat dementia in the past. We're not good at late life medical care in the way that we are at early life medical care, or something like diabetes or cancer. We need to direct a lot more attention, and that means public attention and political attention and financial attention as well to dementia to help find out what it is and what can be done about it. Churchill suffered from depression, and he called this his black dog. Now, I'm afraid dementia is more like a black elephant. It's a very large and threatening object. It's a disease, it's progressive, it's degenerative, and it's going to get bigger as the population ages. So it's something that we need to start paying attention to urgently and properly. And the people at QBI are doing exactly that and deserve our admiration and our support. Last thought. You might remember a film called Dead Poets Society with Robin Williams. And he told his students carpe diem, which they translated in the film as grasp the day. That was a good mantra, but it actually was a bad translation. Because in Latin, carpe doesn't mean grasp, but it means something like pluck and cherish. It's much more gentle than that. And so whether your day is with dementia, living with it, whether it is caring for someone with it, whether it is working on it, every moment in these days are worth cherishing. And that's a good word to finish with. Yeah. Thank you.
Thank you, Rolly, and thank you, panel. Look, what an interesting evening, and you've heard perspectives from lived experience to how we work in the community and what we can do at the protein level. And really, what's needed is a sort of overarching view of it, right from the basic science up to what's happening in the community. And in many ways, QBI is working at all those levels. And look, to have an institute like this in Brisbane is fantastic. So come and visit us if anybody's interested to talk to some of the people that are here and many of the younger people we have. We do have a lot younger people at QBI, who are the next generation. And it's these lot who are really going to find the discoveries and the cures. I mean, honestly, what Stephen said is exactly right. It's going to take time, and it's not folk like us, it's the younger kids who are there. So come and talk to them and, be enhanced and learn about it and see what a wonderful place we have here in Brisbane. So look, thank you. Join me in thanking the panel again. And come and join us for a drink outside. Good evening.